next two sessions, I want to talk about the different patterns of infection that we encounter when viruses enter the host. And today I want to start with acute infections. This first slide diagrams the general patterns of infection that I want to go over. What we're looking at is on the y-axis virus production, also shown as the blue line, and the x-axis is time after infection. And at the very top you see the pattern for an acute infection, a very defined period of virus production, virus ramping to a peak and then declining. The red bar indicates viral disease, and that's the hallmark of an acute infection, a very specific time of virus production, disease, and then resolution. Rhinoviruses, rotaviruses, influenza viruses, and others are examples of viruses that cause acute infections. Next time we'll be talking about the different kinds of infections that don't have a defined start and end. They last for the life of the host, persistent infections. We'll talk about that on Wednesday. Acute infections are characterized by rapid onset of viral reproduction, a short but maybe severe course of disease, so it can be severe disease, intermediate disease, maybe no disease at all, and just virus replication, and an asymptomatic infection, production of large numbers of virus particles, and eventually immune clearance and resolution. You either resolve the infection and survive, or unfortunately in some cases the host dies. Here's another graph showing the course of a typical acute infection. Remember, the characteristics are rapid and self-limiting infections. Here again we're looking at virus growth on the y-axis. On the x-axis we've infected a host, and we're looking at time after infection. There's a period of time when virus numbers begin to rise. Above a certain point, innate defenses are engaged. And then if the virus titer continues to rise, the adaptive response is engaged. Eventually virus production peaks and begins to decline, and eventually virus is cleared. The induction of adaptive responses, of course, results in the generation of memory, so that if this virus is encountered again, we'll have a more rapid response that prevents infection. An important concept for all infections, acute or persistent, is the incubation period, which I define as the initial period before symptoms of disease are obvious. During the incubation period, viral genomes are replicating, the host is responding. So there are clearly signs of illness. Signs are the things you need to measure to be able to find out. The person can't tell you about the signs of illness, but we can certainly measure virus reproduction and the response of the host. So during the incubation period, we have signs of illness as shown in the graph. Genomes are replicating, the host responding. The virus may or may not be transmitted in the incubation period. It depends on the virus. For some viruses, there is transmission during this initial period, but for some others, transmission only occurs when symptoms occur. So for example, Ebola virus is not transmitted unless the person clearly has symptoms of Ebola virus infection. Here are some incubation periods of viral infections, and you can see they range from very short, a day or two, to very long, 50 to 150 days. We generally think that short incubation periods mean that the virus enters, it replicates at the site or near the site of entry, and that's where the primary symptoms are produced. Long incubation periods may mean that the virus has to move somewhere else in the host from the site of entry in order to cause disease. That's certainly characteristic of some of these infections. However, not all of them. The papillomaviruses that cause warts have an incubation period of 50 to 150 days, yet the wart typically forms at the site of entry in the host. For example, the wart you pick up in the locker room on the bottom of your foot because you're not wearing flip-flops, that virus that enters an abrasion on the bottom of your foot typically forms a wart at the site of entry. So there are always exceptions to these rules that we make. Now on this table, the incubation period in days has a letter A that refers to until first appearance of prodromal symptoms. What is a prodromal symptom? Well, that is defined as the period of symptoms before those characteristic of the disease. For example, a characteristic of measles is the formation of a rash. So the time in infection up until that point is the prodrome. It comes from a Greek word prodromos, meaning precursor. There are also inapparent acute infections. Not every infection may give symptoms of disease. If the virus is multiplying, of course, it will make signs of disease. Inapparent acute infections are successful infections with no symptoms or disease. In these infections, sufficient virus particles are made so that they can be shed and spread in the population. How do we know that inapparent acute infections occur? Well, because we can do serological surveys where we take a population and take serum from them and ask, are there antibodies to particular viruses in them? And we can see that people have had infections with a variety of viruses without reporting any symptoms. For example, they have measles antibodies in the blood, but they never had the rash of measles. This is characteristic of what we call well-adapted pathogens. They've been in the population for so long that they don't cause a lot of acute disease. An example is poliovirus, where over 90% of infections are inapparent. They don't cause any symptoms of infection. Acute infections are common public health problems because not only are they the causative agents of serious epidemics that affect millions of people each year, for example, influenza virus and norovirus, but they're difficult problems to control. By the time the infected person feels ill, the infection may be over and has probably spread to others. These are so rapidly developing that unless we're able to diagnose them very quickly, which we can't do at the moment, by the time someone feels sick enough to seek medical care, it's too late to do anything about it. First question, which of the following do acute infections and incubation periods have in common? The virus is not replicating, no symptoms are visible, immune defenses are engaged, the immune system does not respond, or all of the above. The answer is C, immune defenses are engaged. Nobody picked virus is not replicating, that's good. No symptoms are visible. At some point in an acute infection, symptoms are visible, right? So that's not right. That's the definition of an incubation period, that's right, but the acute infection itself, no one picked D, and it's not all of the above. A little tricky there, right? Here are the viruses we're going to talk about today. Tough to pick them, but I pick a range of, these are all RNA viruses. Look at that. Maybe I'm a little biased there, but they're all RNA viruses, and they're transmitted in different ways. So that's one variable that I looked at, and it's a range of icosahedral and envelope particles. So let's start with influenza. We're just reaching the end of the flu season now. There are three types of influenza viruses, A, B, and C. They're all enveloped, negative-stranded RNA viruses with a segmented genome. We've talked about their structure, their replication. You should know that well by now. Both A and B viruses cause a similar kind of disease, which we'll talk about. The type C cause mostly inapparent or mild upper respiratory tract illness. And we only vaccinate against A and B. A is the only one that causes pandemic. This is a global outbreak where every country in the world, or many countries, experience an outbreak at the same time. And the hallmark of these viruses is antigenic variation. We talked a little bit about before how the virus is able to change the sequence of its surface protein so it evades any immunity conferred by infection or by vaccines. These viruses are transmitted by droplets produced by coughing, sneezing, or talking. Remember, we talked previously about how when you do these things, you make different sized droplets that can travel a long way depending on their size. Influenza virus will be present in all of them. It can also be transmitted by direct contact with an infected individual, as long as you get some virus-containing fluids, or by contact with a contaminated surface. The virus doesn't last a huge amount of time on surfaces, but if someone's just touched a doorknob and they have mucus on their hands, and you touch it and then touch your eyes or your nose or your mouth, you're going to be infected. There's a wonderful study done a number of years ago, done in Switzerland, where they took Swiss banknotes and they pipetted influenza virus onto it, and they asked, how long does it last on the banknote? So the right country to do the study in, right? Because they use a lot of money in Switzerland. Without mucus, the virus doesn't last more than a few days, but it could last up to 10 or 12 days, I think the number was, if they mix the virus with mucus. So there's something in the mucus that can stabilize infectivity. And this dried out virus on a banknote. So dollar bills, banknotes, are pretty dirty after touching them, which no one should do anymore. There's no need to, right? You should not put your hands in your eyes or your mouth for sure. You should wash them. So the virus enters the upper respiratory tract by the ways we talked about. The virus replicates in the mucosal epithelial cells and spreads locally within the respiratory tract. No viremia. This is a virus, at least human influenza viruses stay within the tract. It's not the case for all viruses, especially in other animals. Virus replicates in these epithelial cells, which we've described before, and the virus can replicate in the upper tract. So mild infections may just have upper tract replication, but it will also spread down into the trachea, and that's when, in many cases of influenza, you feel pressure in your chest because the virus is replicating there. And if it goes down into the lower parts of the lung, it can infect the bronchi, the bronchioles, more serious result, and of course, the alveoli, that would result in primary viral pneumonia, which is a very serious illness. But again, restricted to the tract. Now, the receptors for this virus, remember, are sialic acids, and human strains of influenza virus interact with alpha-2,6 linked sialic acids, and avian strains with alpha-2,3. Now, if you look at any scientific publication or even the news from time to time, well, in fact, this past Sunday, if you looked at the opinion section of the New York Times, there's an article by Michael Osterholm about how viruses are the real wars that we're fighting, and we shouldn't be raising money for military without raising money for fighting microbes. I think it was called the microbe wars. And he used influenza virus as one of the examples because there are lots of strains circulating in birds that periodically infect people and can be very serious. These avian strains bind alpha-2,3 sialic acids. Now, this is a picture of a human showing you where in the respiratory tract the sialic acids of the different kinds are. So if you look in the nasal mucosa, paranasal sinuses, pharynx, larynx, trachea, up to there, all alpha-2,6, so only human strains are going to infect by those routes. If you're going to be infected by an avian strain, it has to get into the trachea directly by deep inhalation. So a lot of these people who get avian strains are working with chickens, for example, in bird markets, and they're very close to them. Some of them sleep with them at night, and they inhale very deeply. So we think the virus gets into the trachea or even down lower. You can see the bronchus, the alveoli, bronchioles, they all have alpha-2,3. However, there's one other place we think these avian viruses can get in, and that's by infecting the cornea and the conjunctiva of the eye, which have alpha-2,3 linked sialic acids. What good is that going to do you? Well, there's this thing called the nasolacrimal duct, which drains the eye into your nose, and the viruses could, in theory, drain from the eye into your upper tract and then down into your lung. And there have been some outbreaks of avian influenza viruses in people where it's associated with conjunctivitis, inflammation of the eye, and that's consistent with that kind of spread. Uncomplicated influenza is a pretty straightforward disease, short incubation period, abrupt onset. Most people know exactly when they started to feel badly. Headache, chills, dry cough, high fever, muscle aches, malaise, anorexia. Pretty high fever peak within a day, and then it begins to go down. This whole pattern is very different in children and in older people who are much more susceptible to infection. Fever goes down, then you start to have worse respiratory symptoms. You start to make a productive cough, which means you're coughing up mucus, fluids, and this cough and weakness can persist for up to 2 weeks. It's really a debilitating respiratory infection compared to the common cold, where it's over in a few days and you pretty much can get back to normal. And as I've said, the virus can replicate throughout the respiratory tract depending on the receptors for the particular strains. How do you diagnose influenza? Well, in a flu season, which, from the late fall to April, May-ish, it's called an influenza-like illness. And if you have a fever of at least 100 degrees, a cough or a sore throat, and no other cause, the doc will say you have flu without even diagnosing it in any other way, because it's likely that you do have flu, and they may give you antivirals to try and resolve the infection. There are rapid lab tests. You can go to a doctor's office, give him or her some sputum or nasal wash, and they can do a rapid lab test right there in the office, looking for evidence of infection. But these have very poor accuracy. They're wrong almost as much as they're right. And so that's why if you have these symptoms, they'll just treat you right off the bat. There are other ways to diagnose, including PCR, viral culture, serology, but these take much longer. And by the time they're done, you're going to start getting better. And you only have about a 2-day window to give antivirals to treat this infection. So here are 2 curves which were done with volunteers. You can take certain strains of flu in immunized people, and you can do challenge experiments, and that's what was done here. So we're looking at inoculation intranasally with virus, and then we're looking at viral titers in this first experiment, and symptoms. They asked them how they feel on a scale of 1 to whatever in a variety of ways, and they come up with a score here. So you can see right away, the first day after inoculation, you begin to shed virus in the nasal secretions, then there's some symptoms, and they begin to rise. They peak at day 1.5 to 2.5. You can see very rapid peaking and then rapid decline. In this case, the virus is declining as the symptoms are declining. On the graph on the right, similar kind of challenge experiment. We inoculate people. In this case, we're again looking at virus, peaking in this one at 2 days, very rapid peak of virus, and you see, and you've got symptoms throughout these subsequent days, but the virus is declining. Interferon peaks very quickly and then declines rapidly. And look at these other lines, the dotted lines. These are antibodies in the nasal wash or in serum. You can see that these are taking a very long time to rise and are really rising much too late to do much for infection. It's rather probably cytotoxic T cells that are helping to resolve this infection. But you will have immune memory after this. Now, influenza is, you have fever, by the way, in addition to that cough, sore throat? No? Influenza is a seasonal disease. Right? This is the season now in the upper hemisphere, and you can see that on this chart. CDC and WHO has a global network of laboratories, thousands of laboratories that get samples from people with suspected flu, and they send them into regional labs, and they do analysis to find out what strains are there and so forth, because this is a serious disease, and put a lot of resources into it. So you can see here the number of isolates or percent positive, 2 different ways of looking at the data, in 4 different flu seasons from 2004 to 2007. And you can see the A viruses and the B viruses are in different colors. But the point I want to make is that there is a very specific peak of infections, no matter what parameter you look at here, which is occurring towards the end of one year and the beginning of the next, November, December, January, February. These are weeks of the year, by the way, so 4 is week 4, 14, and so forth. So a very clear seasonality in temperate climates. That is our kind of climate where we have a summer and a winter. These are the flu results for this year. So we are approaching, so the way these numberings work is you have the year first and then the week. And so this year's flu season, we're looking at the A and the B viruses here in 2 different colors. You can see began to rise around week, let's look at a slow rise at the end of 2016, starting I guess at about week 44 or so, and then it peaks just around the end of the year, in January or so, week 2, week 4, peaking in about week 8 here, 2nd month February, and now we are in March, so week 12 would be the end of March. We don't have those data yet, but the week before, you can see it's really declining now, and it's going to keep declining until the end of April, roughly. So as I said, we keep a lot of statistics for influenza because it's a serious disease. Another way that we report influenza information is by pneumonia and influenza mortality. So people who die of pneumonia and influenza. Frequently, the most deaths from flu are by pneumonia in elderly or very young kids. And usually on the death certificate it says pneumonia. Very rarely says flu and pneumonia. So they lump the 2 together to get an estimate of the mortality caused by influenza. Of course, if it's bacterial pneumonia, that wouldn't be reported here. But you can see, again, and this is multiple years in the weeks within the year from 2012 to 2017. You can see seasonality of mortality. Now what's done here is they calculate what should be the mortality every year. This is the maximum and the minimum in the 2 black lines. And then each year, it may or may not, what's actually observed may or may not exceed the baseline. So you can see in 2012, 13, and 14, there was major exceeding over the baseline of of pneumonia and flu mortality. Not so bad in 2015-16. This year, not so bad either, just exceeding the threshold. So the threshold is what you would predict in a typical season. For some reason, sometimes there's more mortality for a variety of reasons. It can be the isolate of influenza virus circulating, other conditions as well. So here are some numbers just so you understand what we're dealing with. These are yearly estimates, 35 to 50 million cases a year. We don't obviously diagnose every case of influenza, but we can get a sense by sampling all over the country how many we have, then we can extrapolate. And then in the past 31 years, the number of deaths every year has ranged from 3,000 in a low year to 49,000 in a high year. So that's a lot of people, and especially if you're very young or very old, you should get flu shots because you have more risk of serious disease. But your age as well, you should, because if the more people immunized in a population, you're going to have herd immunity, and that will restrict spread. It's an altruistic thing. Now, whether you're feeling altruistic or not, you should be altruistic in the case of vaccines, because it's really better if other people don't get sick. It costs a lot less for the healthcare system. Now, besides flu, there are complications of this infection, including viral pneumonia when the virus goes down into the alveoli, which is a very serious disease. Secondary bacterial pneumonia, you think you're recovering and all of a sudden you get a bout of pneumonia caused by bacteria that are down there in your lungs. Generalized muscle pain, cardiac involvement. People have trouble breathing because of influenza. It can put a strain on the heart, and if you have a bad heart to begin with, it can cause problems. And then there's a condition called Reye's syndrome, which involves encephalopathy, a swelling of the brain, and liver damage. Not sure how these latter are caused. The virus is not replicating in muscle, it's not replicating in the heart, it's not replicating in the brain or the liver. These are probably secondary effects of cytokines, or in the case of the heart, breathing very heavily. What kind of interventions do we have? You can go to the drugstore and find all kinds of non-prescription or non-pharmaceutical interventions which may or may not work. If you feel better, then it's a placebo effect. That's great. But we do have 3 antiviral drugs. We'll talk about their mechanisms later on when we talk about antivirals. We have Tamiflu or oseltamivir. Here's Tamiflu here. This is a capsule that you take orally. Relenza or zanamivir is an inhaled antiviral. Not everyone can deal with this, especially if you have asthma. You inhale something, it will trigger an asthma attack, so it's not for everyone. They're both neuraminidase inhibitors. And we have vaccines. We have lots of vaccines against influenza viruses, and we will talk about those in some detail in the vaccine lecture. Next question, which of the following is characteristic of uncomplicated flu? Transmission may occur via respiratory droplets. The incubation period 1 to 5 days. Fever peaks within a day. Coughing and weakness can last for 2 weeks. All of the above. All right, as you see, the answer is E, all of the above. Every one of those is something I mentioned. So now that you know the symptoms and signs, did anybody have flu this year? Did anyone get vaccinated? You got flu. Good. Awesome. Very good. Many of you have had influenza and will have it over and over. The next virus, polio, I doubt any of you have had. You might have had an inapparent infection, but none of you look like you have had paralytic polio. And of course, if you were just in the US your entire life, you likely wouldn't have even an inapparent infection. There are only a couple of countries where the virus is still circulating. So here is poliovirus. It's a 30 nanometer icosahedral particle, which we've talked about in terms of its structure. It's had an RNA genome inside of positive polarity, about 7,400 bases in length. And the way you acquire poliovirus is by fecally contaminated material. The world is covered with a thin layer of shit, and a lot of it is human shit. And we often put it in our mouths. We have very bad hand hygiene. Yeah, I'm sorry to tell you that, but anything you touch is fecally contaminated. People have done studies of stuff, and you can find fecal coliforms in so many things. You put the virus in your mouth, and kids are very good at doing this, especially in the summertime. Virus goes into your intestine, passes right through your stomach, low pH digestive enzymes, goes right through it. It's amazingly hardy. goes into the intestine. In some cases, the virus can replicate in the oropharynx if you get enough, and then that virus will go down into the intestine as well, replicate in the mucosa, spread to the local lymph nodes, and then eventually to the blood. Now, in 99% of infections, it ends there. You may have no symptoms whatsoever, or some flu-like symptoms, if you will. And the virus is being excreted vigorously from your GI tract, and that's how it spreads to others. So many people don't even know they're infected. They're shedding virus, and that is effectively spread to other people. In about 1% of people, the virus gets into the central nervous system. It does so from the blood. It can enter the blood, the CNS, in two ways. The virus in the blood will get to the muscle. There are receptors on the ends of nerves in the muscle. These, of course, are innervating the muscle so that it can move. The virus will bind to those receptors. They'll be transported up the nerve by retrograde transport, and eventually reach the cell body of the neuron in the spinal cord. The virus will replicate in it, kill it, and eventually you kill enough neurons and you get paralyzed. The virus can also probably percolate from the capillaries into the spinal cord and the brain as well. So here's a slice of a transgenic mouse spinal cord showing you, this is from an animal that was paralyzed after infection. And all these green dots here, this is poliovirus RNA. So this has been hybridized with an RNA probe to show you where the virus is replicating. These are all neurons, very specific for neurons within the spinal cord. As I said, you destroy enough of them and you get paralysis. And on the right, you can see a single neuron full of viral RNA. Here's a lovely chronology of this infection. This is taken from studies in the 40s and 50s of a number of cases of polio in people. And it's a hand-drawn figure, which was the norm back then because we didn't have computers, so everyone got to be really good at drawing their own figures. And what we have here is an exposure here, and then different days after infection, what's happening in different compartments. So at the top, for example, we have the temperature. So you can see there's about a 7-day incubation period, and then a spike in temperature here associated with headache, sore throat, nausea, flu-like symptoms, if you will. These people get better, and that's the end of their infection. However, they do shed virus in the feces. You can find virus in the throat in these individuals. You can find virus in their blood. You can see on the third panel here. It peaks and goes down, and these individuals have antibodies. Now, in 1 in 100 or so individuals, the infection proceeds into the CNS. Another 5 days of incubation, and then another spike, a higher spike of temperature, accompanied by headache, nausea, stiffness, pain, and in some cases paralysis. So that is full-blown poliomyelitis. And that's accompanied, if you go all the way down to the bottom panel, by virus in the central nervous system. All right, so it gets there in these individuals who get paralyzed. So those who recover probably don't have any virus in their CNS. And why the virus gets in some people is not known. I think it's a polymorphism in some innate immune gene that is making virus replicate more robustly and allowing it to get in the CNS, but nobody's answered that question. People are the only reservoir for this virus, so if we can eradicate it, and it should not come back. As I said, it's spread by fecal-oral transmission. It is seasonal in temperate climates, peaks during the warm month, and that's why in the peak of polio in the US, they didn't want kids going out in the summertime because they thought that's how they got it. There is a long-term complication called post-polio syndrome. 30 to 40 years after you've had polio, you can recover limb function by therapy and have a totally normal life. Some people, 25 to 40% of those individuals, they begin to be paralyzed again. They lose limb function. It's not an infectious process. There's no virus around. It's probably because when they had polio, they lost a lot of motor neurons. And normally, as you age, you also lose motor neurons, and people with polio simply don't have a backup. So they become paralyzed. And there's still people developing this today because that we still had polio in the 60s in the US. Now, there are 2 vaccines that are very effective in preventing polio. We'll talk about these a lot in our vaccine discussion in a week or two. But just to show you, we had 20 to 30,000 cases a year of paralytic polio in the US. in the 20s, 30s, 40s, and 50s, and just multiply that by 100 to find out how many people were actually infected, right? In 1955, the inactivated vaccine was introduced. That brought this down to a few thousand cases a year. And then in 1961, the infectious oral vaccine was introduced. And in 1979, we had the last case of polio in the US. And since then, we have had none. And in many other countries of the world, there is no polio as well. But we'll get into that more later on. All right, that's poliomyelitis, a very classic acute infection. All right, our next acute infection is measles virus. And this is a different virus structurally. It's an envelope virus with a negative strand genome. And it's in the family Paramyxoviridae, one of the most contagious human viruses. The R0, this is a parameter we'll talk about a bit later. The R0 is basically how many people you can infect if you're infected with a virus. On average, how many other people will you infect? So with measles, the number is very high, 15, and very contagious. I always used to say that if we didn't have measles immunity, and I had measles and I were exhaling it, you would all be infected within a few days because it's that contagious. There's one serotype of this virus. By the way, there are 3 of polio. I didn't tell you that, but they all cause polio, and only 1 of measles. And if you get infected, you have lifelong protection. You never get infected again. The virus is transmitted by inhalation of respiratory secretions. Very different from polio, but similar to influenza. And of course, the classic presentation of measles is the skin rash, but you are actually contagious for several days before the rash. So in the old days when we didn't have vaccines, kids would go to school. They didn't feel great, but they went to school, and they're shedding virus, and they spread it because it's respiratory transmitted, very readily transmitted. And then, of course, they would get the rash and go home, but by then they've already infected others. And here we have an interesting situation where almost every infected individual has some sign of disease, some symptom of disease, like a flu-like illness and so forth. Not like polio, where the paralysis is only in 1% of the individuals. Now, this virus enters the upper respiratory tract by aerosols or contact. Where it replicates initially is very interesting. It doesn't replicate in the mucosal epithelial cells, but rather in immune cells that are patrolling the epithelium. And that's shown in this diagram on the right. We have respiratory epithelium, and these little cells, which look like dendritic cells, or other immune cells, they patrol the epithelium. They are the ones that are actually infected with the virus. The respiratory epithelium can't be infected from the apical side. These immune cells go in, they go to the lymph nodes, the virus replicates in lymphoid cells very well, and then the virus goes into the blood and you get the primary viremia. From the blood, the virus goes to secondary organs. One of them can be the brain, so you can have serious brain infections caused by measles virus. And of course, it goes to the skin, where you get the typical rash presented of measles. As the virus is circulating in the blood, various lymphoid cells are infected, and these will at some point go into capillaries that underlie the epithelium of the respiratory tract. These cells will release virus. There are virus receptors on the basolateral surface of these lung epithelial cells. Virus enters the basolateral surface, replicates in them, and is then shed from the apical surface. And that's how the virus is spread to other people. So virus is shed into the lumen of the lungs. You cough or sneeze, and you exhale the virus. Now, this is very different from flu or rhinoviruses, which enter the apical side of the respiratory epithelium. They are shed from the apical side. They don't come out of the basolateral side. They don't enter the circulation. So measles doesn't enter the apical side because these receptors for the virus, Nectin-4 they're called, they're only on the bottom of the cells. They're not on the top. And so people figured this out just a few years ago, that the way the virus gets in and out are very different. So here are the signs and symptoms of measles: high fever, respiratory symptoms, stuffy nose, cough, conjunctivitis, inflammation of the conjunctiva of the eye, the rash, of course, from the face to the extremities, which is shown here, and what are called Koplik spots in the mouth, the buccal mucosa, these white spots here. These are cells infected with the virus, and if you took a little sample of these and looked at it under a microscope, you would see syncytia Remember what those are? Fused cells with lots of nuclei. And if someone came and had these and you looked at it, it's probably measles. Measles is one of the few viruses that does that in the mouth. So it used to be diagnostic a long time ago. There are complications of measles, which is why it's not just a rash disease with some respiratory systems. You can have very serious complications. The virus, as I said, gets into the brain in a fraction of people and can cause acute and post-infectious encephalitis. And that has a fatality rate of 1 in 1,000 kids. Also serious lung diseases, ear infections. Overall, the fatality in the US was 1 to 2 per 1,000 cases when we had lots of measles before the vaccine. It goes up to 28% in countries with poor nutrition, because poor nutrition further suppresses your immune system. A very late sequelae is called subacute sclerosing panencephalitis. And so this is a persistent infection caused by measles. So this virus can do acute or persistent infections. And this is a disease that happens maybe 10 or so years after measles. The kids who are otherwise fine, they start to have their CNS degenerate, and they eventually die. So it's a degenerative CNS disease. There are non-infectious virus particles in the brain, and we're not quite sure how this happens. We'll talk a little bit more about this on Wednesday. And of course, measles virus immunosuppresses, as we talked about last time, and that leads to a number of secondary infections, and it's the main cause of death in third world children. Immunosuppression by measles leading to other infections that kill these kids. So we do have a vaccine for measles because it's a serious disease. In the US, pre-vaccine, these were some numbers. We had 3 to 4 million cases of measles a year. When I was a kid, we just introduced the measles vaccine, but I saw lots of kids with measles, rashes all over their body. 400 to 500 deaths a year, 48,000 hospitalizations, and 1,000 per year kids with chronic disability from encephalitis, from virus going into the brain and causing damage. The transmission in the US was stopped by the vaccine by 2000, and now this virus vaccine is given as a trio with measles, mumps, rubella vaccine together. And you may remember back in 1998, a gastroenterologist by the name of Andrew Wakefield published a paper saying that this vaccine caused autism. And that, of course, led to decreased use of this vaccine. It has been refuted and withdrawn and shown to be wrong over and over and over again. He lost his license in the UK, but he's now in Texas and still doing anti-vaccine activism in Texas. And even though this vaccine is safe, it prevents measles, there are still people who believe him and will not immunize their kids. There have been many outbreaks in the UK and Ireland, and these get imported into the US, and they find the people who aren't immunized, and there are outbreaks in the US. So here's the epi of measles. Lots of cases in the US, the vaccine licensed in the 60s, and as I said, almost eradicated in the US. Every now and then there are blips of outbreaks of people who don't immunize their kids. The inset is deaths per year, so we had lots of deaths per year, and still if there are outbreaks, they're accompanied by deaths because this is a lethal virus. This is a graph of cases by year from 2001 to 2015. Now again, this is a vaccine preventable disease, but every year there's some cases of measles. This is in the US. And you can see sometimes there are big outbreaks, the big one in 2014. And the one in 2015, there were 178 cases in 17 states. And these are all people who did not immunize. These are kids who were not immunized. Their parents chose to not give them vaccine. And they tend to cluster. You know, there's this neighborhood effect where people in a neighborhood will talk, are you giving your kid MMR? No, I don't think so. Yeah, I'm not going to give it either. And you get numbers of kids building up, and boom, the virus comes in, and you have an outbreak. And some states more than others. This is in 2015, where there was a big outbreak in California, started in Disneyland. Because all the unvaccinated kids went to Disneyland, and you put the virus in, and boom, big outbreak. And you can see some other states, Washington, Nevada, Arizona, Illinois, had lots of cases as well. But pretty much these happen in a lot of states. And they could all be preventable by vaccinating their kids, which would have no harm on the kids. So I do not understand why people don't want to vaccinate their kids against this. Now here's the global situation. There's a lot of measles elsewhere because many countries don't get the vaccine. They can't afford it. They would love to have it, believe me. They would love to have the vaccine that people here don't take. But here are the highest numbers. Over 1,000 are the darkest colors. Parts of Africa and Asia still have over 1,000 cases a year because not enough kids are immunized. And many other countries, you can see, have lots of measles as well, either because of anti-vax activism or because the countries don't have good coverage because they can't afford it. There were 114,000 deaths globally in 2014 from measles. These are all preventable. This is inexcusable. From 2000 to 2014, we prevented 17 million deaths by vaccinating. This is a really great public health buy. This is a very worthwhile vaccine, and its trashing is completely unconscionable. All right, next question. Which of the following is a good reason to get measles vaccine? There is a 1 in 1,000 chance of acute post-infection encephalitis. There's a 1 to 2 in 1,000 chance of dying. Each infected person spreads measles to 15 others. Immunosuppression can lead to secondary infections. So all of the above. E is the answer, of course. All of these things are good reasons to get measles vaccine. In a 24-hour period, 200 million people in the world have gastroenteritis, and the amount of water they pass equals what flows over Victoria Falls in 1 minute, which is 65 million liters per minute. That's how much in a 24-hour period is excreted by these people with gastroenteritis. So that's a gastric, an intestinal disease where you pass lots and lots of water. 65 million liters for a 24-hour period. So a gastroenterologist gave me this slide a couple of years ago. I love it. Biggest cause of gastroenteritis is norovirus, which is a small RNA virus. It's a plus-stranded RNA virus in the family Calliciviridae. It's built with icosahedral symmetry. It causes half of all foodborne outbreaks of gastroenteritis, 23 million per year in the US, and that's probably an underestimate. And after I talk to you, you won't eat anymore. Right? And you won't go to any parties, and you won't go to any cruises or anything like that. Actually, it's not a serious disease if you don't mind vomiting and having diarrhea for 3 days. So here are the known causes of foodborne illnesses in the U.S. for the period 2006 to 2006. Norovirus, 49%, bacteria, 40%, and then some other causes here. You can see an estimated number of illnesses was 5.5 million for norovirus, and then a variety of bacteria. We once thought that bacteria were causing most of the gastroenteritis until we discovered noroviruses and rotaviruses, which are two major causes of this illness. And so there's lots of effort to understand how this works. This is a two-bucket disease, as I've mentioned before. You are vomiting and you are having diarrhea. And it's spread fecal-orally, of course, fecally contaminated material. The virus passes through the stomach very much like poliovirus. It then replicates in the mucosum. Actually, it replicates in the GI tract, probably not the mucosal surfaces, as you'll see. This has changed in the last year. And one of the effects is blunting of villi. Here we have proximal jejunum sections from that part of the small intestine. And this is a normal villi on the left here. And these are from an individual with norovirus gastroenteritis. So the villi are blunted. We don't know really why it causes vomiting and diarrhea, something the virus is doing. It might be making an enterotoxin that causes the diarrhea, for example. People are working on understanding that, but we don't know at the moment. What we do know that's very interesting and has informed our understanding of norovirus is that bacteria in your gut promote replication of this virus in B cells. And this applies to humans as well. But this panel is with mouse norovirus. So what we're looking at here is two different experiments. We're looking at virus replication on the y-axis, days post-infection. And these are cultured B cells, and they're infected with a norovirus preparation which has been filtered to remove bacteria or unfiltered. And you can see the filtered preparation, there's very little virus replication, whereas the unfiltered prep, there's lots of replication. So why did people even look at this? Well, if you remember, I told you that you can use people as volunteers and give them norovirus. What you do is feed them a little bit of stool filtrate from a person who has norovirus gastroenteritis. And if you want to study the virus in the lab, you also take these stool filtrates and put them on cells because you couldn't grow the virus up until recently. So you filter them to remove the bacteria and all the other material in the stool, and then you try and work with them. And for years, no one could get human noroviruses to grow in cell culture. And one day, they forgot to filter the stool extract and put it onto cells with the bacteria from the stool in it, and it grew. And that's what this virus, this graph is showing you. So you need bacteria in order for norovirus to replicate, probably in the gut as well. Here's what we think is happening. This is a figure from a paper that just came out in the past year. Why do you need bacteria? Well, it turns out that there are carbohydrates in the outer surfaces of bacteria that the virus binds to. And in your gut, here's your gut with the intestinal epithelial layer. The norovirus shown here in red, and this is a hypothesis, of course. We don't know if it's all correct. We know that the virus will bind bacteria and components of bacteria, and that allows it to be infectious in cell culture. We think the bacteria then transcytosed into the subepithelial layers here, and there the noroviruses replicate in B cells. Now, how did the bacteria get through? We're showing two ways here. So here is a bacterium attached to a norovirus. We're showing it get through an M cell. M cells, remember, are very good at taking material from the gut lumen and transcytosing it across into the subepithelial space. So that's one possibility. The other is that the virus can get through normal epithelial cells as well as a passenger on the bacterium. So the virus gets into the subepithelial space and then replicates in B cells, which are these cells with the big nuclei here. Now, why this replication causes blunting of the villi is not clear, why there's diarrhea is not clear, especially since the virus is replicating in this subepithelial space. It's some really interesting questions that a number of people are trying to sort out. But now you can grow human noroviruses in B cells in culture as long as you add components from bacteria, and that seems to enable the viruses to get into cells. Now this is, like measles, a very contagious virus. 18 particles have been shown to be enough to infect a human. They've done dose-response studies, giving people small and more amounts of particles, and 18 is enough to get you sick. And this is a lovely factoid. The amount of virus on the head of a pin could infect more than 1,000 people, in theory. Except I don't know people that eat pins, right? So I'm not sure that's going to work. Some of the features of infection: fecal-oral transmission, for sure, contaminated foods and so forth. We'll talk about that in a moment. But also aerosols. People vomiting, they create an aerosol. That can go in your mouth. Yeah, it can. And you can swallow it and acquire norovirus infection that way. Also, contaminated materials can have the virus on. It's very stable on surfaces. Food can have it, water, environmental contaminate. Food can be contaminated at the source. So oysters, raspberries. Oysters can be filtering contaminated waters. Raspberries can be washed with contaminated water that has fecal material in it. And that you eat those, you will get infected. Or doing preparation by food handlers. Often food handlers are excreting and not sick. So they contaminate food and they spread infection. Pretty short incubation period, sudden onset of vomiting and diarrhea, stomach pain. Pretty short illness, 28 to 60 hours. 30% asymptomatic infections. So 30% of people are infected and shedding and not having any symptoms, so they're going to go to work. And if they're food handlers, that's a big problem. And so that can be, a food bar or a cruise ship or any kind of gathering where there are people taking care of the food prep. They can contaminate it. This virus infects all ages, and it is found year-round. It does peak in cold weather, but you can find neuroinfections all year round. And the outbreaks often occur in semi-closed environments, like nursing homes, hospitals, cruise ships, all these places where lots of people gather together, and you can get good person-to-person spread. And these are things that we've done more of in the last 40 years or so, nursing homes, daycare centers, and so forth. We didn't have so many of those many years ago, and so the the proliferation of these has also caused an increase in the cases of norovirus. The virus shedding peaks a couple of days after illness, but it can persist for 56 days. Can you imagine? So you feel better, you go back to work, you're still shedding virus. And this is why in every restaurant bathroom there's a sign that says employees must wash their hands. Every time I see it, I think norovirus, and that's why, because that's the main virus that they're afraid of, the main pathogen. They're going to transmit it. So if they don't wash their hands, they're going to have fecal material, they're going to go back into the kitchen and contaminate food and infect people. The sign's not for you, it's for employees. But you should wash your hands anyway. Now the problem with noroviruses is that immunity after infection is short-term. There are lots of different types, genotypes circulating globally, and so you tend to get reinfected after so many months. Your mucosal immunity, which is the most important in protecting you, declines, and you can get infected over and over. Now, people are working on vaccines. It's going to be a kind of tricky issue to sort out with vaccines. The only thing you can do is to prevent dehydration. Take fluids, and that will get you through it. Humans seem to be the only reservoir, though there's some idea that there are some animal reservoirs involved. Some marine mammals seem to be infected with noroviruses. Remember the very first lecture, I showed you a picture of a whale, and I said it was shedding 10 to the 13th caliciviruses a day. That's the same family as noroviruses, and there's some evidence that sea mammals in areas close to the shore can sometimes infect people. As I said, we're making vaccines of various sorts, which will be good for, I don't know if everyone needs to get it, but certainly people who are food handlers should get this vaccine to prevent spread of the virus. So these are not serious infections, as I've said. If you want to take your risks, you can, but if you do have immune-compromised disease, you should not, or underlying illness, you should not take the risk. So here are some settings and modes of transmission which are interesting. So settings where transmission occurs with noro, 40% restaurants and catered meals. So again, people who are shedding virus. Retirement centers, nursing homes, hospitals, schools, daycare centers, vacation setting, including cruise ships. So this is not the majority, even though you hear about cruise ships all the time. That is not the majority of the infections. And the modes of transmission, mostly person to person. That means a food handler contaminates someone else, but foodborne is only 10%, even though it's a major source of infection. Now, a couple of interesting stories here. There was a story on an airplane a number of years ago. Someone vomited. They had norovirus gastroenteritis. So you could board a plane and feel great, and in the middle of the flight, depending on how long it is, you could develop the two-bucket disease and start vomiting. And of course, they have those bags, but you're still generating an aerosol. And what happened here is that the flight crew helped to clean up, and then they all went on a different flight, and people on those flights got sick. So the flight crew got infected, and then they spread it to other flights as well. So this is very contagious, very contagious. And then, of course, there's cruise ships, the happiest place on earth. As I showed you in the statistics, it's not a huge number, but these make the news because these are huge ships. If you've ever been to Miami, you look out at 5 p.m., they're just parading out of the harbor there. And they're huge, thousands of people. There are outbreaks, and they're serious because the ship has to go back and get everyone off, and they have to decontaminate the whole thing, which is really serious. So they follow them. These are just a number of cruises where there were outbreaks, and sometimes they can tell it's norovirus, but sometimes it's something else. There are other viruses out there that are causing outbreaks on cruise ships. Now, we got a letter from a guy who works for the Norwegian lines, and he's the ship's agent, and he said, we have tens of thousands of Purell dispensers in all of our ships. You know, it's that goop with 70% ethanol in it. He wrote us a letter on Twiv. He was very proud of it because he's a listener, and he hears us talk about norovirus, and we said, it doesn't do anything for norovirus. It will not be inactivated by 70% ethanol. And then about 2 years later, he wrote back and he said, okay, we have now installed these high-tech hand washing stations on our ships. They're basically, you go in the bathroom and they're holes, you put your hands in, and it washes them and dries them for you automatically. So that'll take care of the norovirus. So he listened to us. So this is a big deal. because it's an economic thing. But as I said, it's not the major part of how the virus is transmitted. But health officials do track illnesses on cruise ships, so we find them more often than on land. Close living quarters, of course, in a cruise ship, and new people arriving can bring virus in and contaminate surfaces and the crew as well. So how to protect yourself: wash your hands, rinse your fruits and vegetables because the virus can be on them. It can be contaminated at the source. Shellfish, cook it, because the shellfish are filter feeders and they're going to take norovirus in from the ocean or the water. Clean things, wash your clothes, and if you're sick, don't prepare food or care for others. And most of all, don't eat the mince in the restaurants on the way out. Even if they have a little cellophane pack, they're still fecally contaminated, okay? People come out of the restroom, they don't wash their hands, or if they do, they don't do a good job. And people have done surveys of these mints. They're contaminated with fecal coliforms. The world is covered with shit, and most of it's human. So please do not eat them. And if you do and you get sick, let me know. I'd love to hear about it, so I can tell another class about your experience. Okay, the last virus is West Nile virus, which I love in this New Yorker cartoon. But it has nothing to do with Egypt. It's actually first identified in the West Nile district of Uganda, okay, which is not Egypt. And so the Shanahan doesn't know what he's doing here, but it's funny anyway. West Nile is a member of the Flaviviridae, first isolated in 1937 in the West Nile district of Uganda, not the Nile of Egypt. Anyway, we didn't have it in the Western Hemisphere. It was only infecting other Asia and Africa, European countries. 1999, it came to the US, and here it came to New York City. In the summer of '99, there were crows dying in New York, and in the Bronx Zoo, a lot of the exotic birds started dying, and at the same time, people in Queens started having encephalitis, which was caused by infection. It turns out that the virus in New York is almost identical, probably identical to a virus previously isolated from a goose in Israel, so extensive West Nile infection in those countries. So probably came here either in a mosquito in a person on a transatlantic flight and then took off in New York. Why that particular time is an interesting question. Besides humans, it infects hundreds of different birds, mosquitoes, and 18 other vertebrates. This is the global distribution of West Nile now in green, and it's all the US, North America as well. It's overlapping with St. Louis, so that's why the color is in green there. So it's a flavivirus, which you've encountered before, enveloped plus stranded RNA viruses, same family as Zika virus, which we'll talk about in another time in this course. And here's the transmission cycle. So the virus is maintained in wild animals by mosquitoes. So here is the bird transmission cycle. Many birds are natural hosts for infection. Viruses are picked up from the bird by a mosquito. They bring it to a new one. The mosquito is fine. The virus replicates in the mosquito. The mosquito doesn't get sick. And the birds, if these are wild birds that have been living with the virus all their lives, they're fine as well. The problem is when the virus came to New York, the crows had never seen it before, so they got sick. The only birds in the zoo that got sick were the exotic birds that had never seen the virus before in their history. But there were many bird species in the zoo that came from places where West Nile is endemic, and they were fine. Very interesting story. I talked with the director of the zoo last year, and they have all these wonderful samples archived forever that can be studied up there. Really interesting story. Now, the mosquitoes, when they encounter a human or a horse or a number of other species, will bite them, transmit the infection. It's a dead end. It doesn't spread from a human to a human. It spreads from birds to people. So people and horses are dead ends, and because they're new, they haven't evolved forever with the virus, they get sick. And that's the problem with this disease. So it's transmitted by culicine mosquitoes, 3 to 14 day incubation period, and about 20 to 30% of people develop a flu-like illness that we call West Nile fever, not a serious disease. The rest have no symptoms whatsoever. But of these 20 to 30% that are infected, a number of individuals, 1 in 150, develop neuroinvasive disease. And this is why we worry about West Nile virus. This involves headache, eye manifestations, muscle weakness, cognitive impairment. paralysis that looks like polio, 10% mortality. And over half of these people where they have neuroinvasive disease defined by these symptoms, they have long-term neurological sequelae. They have motor issues, they have cognitive issues. So this is not a good thing. In general, it is not a good thing to have virus replicating in your brain. So the way the pathogenesis of infection works, so here on the top is our bird that has West Nile virus in it. A mosquito picks it up, bites a human, delivers the virus into the skin. And remember, in the skin are a number of different cell types. One of them is a Langerhans dendritic cell, which is patrolling the skin looking for foreign antigens. It turns out the virus will replicate quite well in these cells. The Langerhans cells, when infected, go to the local lymph node. The virus can replicate in the lymph node in various lymphoid cells, and then it establishes a viremia from which it goes to other organs, other lymphoid organs like the spleen, full of lymph cells of various sorts, and the virus replicates there as well. The virus can also enter the brain in a fraction of individuals, as I just told you. And last time we talked about how TNF-alpha produced when toll-like receptor 3 senses the presence of West Nile virus. That loosens up the blood-brain barrier, allows the virus to enter the brain. So below here is the brain. You can see a neuron there. And the target of the virus in the brain is the neuron. The virus replicates in it, and that's why you have paralysis, cognitive problems, and so forth. The virus clearing of infection depends on a number of immune types of cells and molecules, including a variety of T cells, B cells, and cytokines as shown here. So recovery from infection depends on these, has been shown in a number of experimental situations. So in the USA, from 1999, when the virus first came in, to 2013, the number of cases varies each year. You can see some years there are 10,000 cases. Other years there are a few thousand. Other years there are fewer cases. And on the bottom, we've broken them down into those with neuroinvasion. So thousands of people each year can have neuroinvasive disease. And we can have deaths as well, as shown here at the bottom. Now, why it varies each year is not known, but one of the parameters that controls it seems to be the overall temperature. In hotter and drier summers, there seems to be more West Nile activity. And one of the theories is that when the weather is hot, the birds go away from urban areas like this. And so mosquitoes have people to bite instead of birds, because otherwise they prefer to bite birds. But if there are no birds around because they've left because there's no water, then they bite people. But that's a theory, and it's not clear if it's correct. This is a seasonal disease. This is the West Nile cases from 1999 to 2015 reported to the CDC in the US according to the week of onset. And you can see this begins in May, June, goes through the summer into the fall and stops at November. This is roughly the mosquito activity time. So mosquitoes begin to be active in the spring there, and they're active through the summer into October or so, depending on where you are. Here in New York City, I heard a talk a couple of weeks ago by a New York City entomologist, and he said we have lots of culisine mosquitoes, which are the ones that transmit West Nile virus, and they start in May and they go through October in New York City. We have no Aedes aegypti here in New York City, and he said we haven't had it for 50 years, which of course is the mosquito that carries Zika virus. Now, if you look at where in the US these neuroinvasive cases occur, this is just a barometer for infection in general. You can see by county over that 16-year period, most of them cluster in areas of the US where there's lots of mosquito activity, as you might expect. Many other counties do have activity. We have West Nile here in the tri-state area, but it's simply not as numerous as elsewhere because our mosquito season is restricted. Now, the disease, the neuroinvasive disease, is more prevalent. It increases in prevalence as age increases. So it tends to be very low at a young age and increases with all of these age groups until you can see greater than 70 years of age. We have a higher incidence per 100,000 population. So this increase in neuroinvasive disease, as with age, probably has to do with losing the ability to prevent invasion of the CNS, reduced immune defenses, and so forth as you age. Now, how do you prevent infection? We do have a number of vaccines in development now, very many actually. If you go to clinicaltrials.gov and search for West Nile virus, you'll find over 10 different vaccines in various stages of testing, and they can be tested for efficacy in the US because we do have enough West Nile disease. So at some point there will be a vaccine for this. So if you're someone who has a lot of contact with mosquitoes, if you want to go camping in the summer, I would get this vaccine because you don't want to get neuroinvasive CNS disease. You might as well get the vaccine. In the meanwhile, you have to be smart about your exposure to mosquitoes until we have a vaccine. These species bite from the evening to the morning. So if you're going to be out and about at your campsite, you have to take precautions to reduce the likelihood that you're going to be bitten. There are very good ways to do that. There are lots of safe repellents that you can use. You can dress properly so that you don't have too much exposed skin and you're not going to be bitten by a mosquito. And of course, in your home, to prevent being bitten, you should have screens on your windows. Of course, in the US, many people have air conditioning, so that is not an issue. But in many parts of the world, there's no AC, there are no screens. When people want to cool off, they sit out on the porch, and that's when they can get infected. But as I said, we have some vaccines which will eventually solve these issues for those who take them. So let me end up with some thoughts about viruses in the CNS. We've talked about polio, measles, and West Nile getting into the central nervous system. All of these viruses are very effectively transmitted by shedding, like polio, measles virus in the gut or the respiratory tract, or by mosquitoes for West Nile virus. That's the main way that these viruses are transmitted. And when you think about it, once a virus gets into the CNS, it's not going anywhere, at least in humans. In animals, you can imagine animals eat each other and they would get a brain virus and that would transmit it. But in people, the CNS is a dead end. So I think, in general, CNS invasion is a dead end in people. It's an accident of infection. There happen to be pathways into the CNS, and these viruses it's very rare for viruses to get in there, and once they get in there, they do cause disease, but there's no evolutionary selection for a virus to get into the CNS, if you think about it. The only selection, in my view, the main selection, is for transmission to a new host. That is what drives the evolution of viruses in populations. So a virus going into the CNS is not selected for. So I say this to you, I've thought about this for many years, because whenever a new virus emerges that causes a new disease, people immediately say this has arisen by mutation. And Zika is the latest example. Suddenly has this property of invading the CNS of fetuses and adults as well. And this is certainly not something that was selected for. All right, it was either present since the beginning and we never saw it, or it was selected as a consequence of some other mutation that arose. There is no way that viruses are selected for to replicate in the CNS, because these viruses wouldn't go anywhere from there, and they're beautifully transmitted by other routes as well.